Welcome to this introductory lecture of Neuroanatomy. In this section, we will explore the fundamental principles that every student needs before diving into the complexity of neuroanatomy. We'll review the basic organization of the nervous system, its major components, and the essential concepts that will guide us through the rest of the course. I hope you enjoy this journey into neuroanatomy and that it becomes as fascinating for you as it is for me. Let's get started. The first thing we need to understand is that there are two main ways to approach the study of neuroanatomy. One is a purely structural perspective, and the other is a functional perspective. To put this into context, imagine that structural neuroanatomy is like studying the hardware of a computer, the keyboard, the mouse, the monitor, everything you can see, touch, and physically manipulate. These are the tangible components of the system. On the other hand, studying neuroanatomy from a functional perspective is like studying the software of a computer. Here, we focus on the logical processes and mechanisms that allow the system to operate. So, in the structural approach, we explore the physical components of the nervous system, and in the functional approach, we focus on how these components interact, communicate, and produce function. It's important to emphasize that these two perspectives, structural and functional, are not opposing approaches, they complement each other. When we study both together, as we will throughout this course, we gain a much deeper and more complete understanding of neuroanatomy. The structures make sense because we understand their functions, and the functions become clearer because we know the underlying anatomy. By integrating both perspectives, you'll be able to appreciate the nervous system as a unified, coherent whole. Now, let's talk about how the nervous system is divided according to each perspective. From the structural approach, the nervous system is divided into the central nervous system and the peripheral nervous system. From the functional approach, the nervous system is organized into the somatic nervous system and the autonomic nervous system. For now, we will begin by studying the structural approach, as it provides the foundational framework we need before moving on to function. There is an important point we need to clarify. In many textbooks, the functional approach, meaning the somatic and autonomic nervous systems, is presented as if it were a subdivision of the peripheral nervous system. However, this is only partially correct, and in many ways, misleading. The reason is this: in the structural approach, we divide the nervous system into the central nervous system (CNS) and the peripheral nervous system (PNS) based on anatomy. But both of these structural components contain elements that participate in somatic and autonomic functions. For example, several nuclei and pathways within the CNS are essential for autonomic regulation, and others are involved in somatic motor control. Likewise, the PNS contains peripheral nerves that carry both somatic and autonomic fibers. So, placing the somatic and autonomic systems as subdivisions only of the PNS ignores the fact that their functional components are distributed across both the CNS and PNS. Understanding this distinction will help you develop a more accurate and integrated view of how the nervous system is organized. Here we can see, according to the structural approach, the main nervous structures that make up the central nervous system and the peripheral nervous system. This distinction is actually very simple. Structures that belong to the central nervous system are located along the midline of the body. These include the brain and the spinal cord. In contrast, the structures of the peripheral nervous system are those that extend outward from the midline, such as the cranial nerves, spinal nerves, and their branches. Now things get a bit more complex. Within the structural approach, both the central nervous system and the peripheral nervous system are further divided into multiple regions and eventually into many individual structures. Each of these structures will be studied in detail later in the course, where we'll explore both their anatomical characteristics and their functional importance. So in this class, we won't go deep into any of them yet. Instead, we'll keep a broad, general perspective. You will have dedicated lessons focused specifically on each structure. What matters at this stage is that this diagram helps you locate these structures in space to recognize which ones are above or below others, which are lateral, medial, anterior, or posterior. The goal is simply to give you a spatial context so that, when we study each structure in detail later, you already know where it belongs within the nervous system. So far, we've been exploring the macroscopic side of the structural approach to the nervous system. Now it's time to move into the microscopic level, that is, to understand what these tissues are actually made of. And, as I'm sure you already know, they are made of cells. Among them, the most important are the neurons, which are the primary and functional units of the nervous system. But neurons are not alone. We also have neuroglial cells, which play crucial supportive, metabolic, and protective roles. Here, I'm also showing you a diagram that compares the glial cells of the central nervous system and the peripheral nervous system. Even though the list of cell types may look long, many of them perform analogous functions in each system. For example, oligodendrocytes in the CNS and Schwann cells in the PNS both serve the essential role of producing myelin. Recognizing these parallels makes it much easier to understand glial cells across both systems, although this is, of course, a broad generalization that can oversimplify the unique functional identity of each cell type. For our purposes here, it's more than enough to build a clear and practical foundation. Let's begin with neurons. These are the functional cells of the nervous system, and for one simple reason, they are excitable cells. This means they can receive and transmit electrical impulses, which is what makes them so special. In this diagram, you can see a typical neuron, and we're going to review its main parts and what each of them does. First, we have the dendrites. These structures receive incoming signals from other neurons, usually from the telodendrion of a presynaptic neuron. Next, we find the cell body, or soma, which contains most of the cytoplasm and the nucleus. This is where the dendrites emerge from, and it is also where the axon begins. The axon starts at the axon hillock and continues through the axon initial segment, which is the region where action potentials are generated. These electrical impulses then travel along the length of the axon until they reach the telodendrion, the terminal branches of the neuron. Through the telodendrion, the neuron forms synapses most commonly with the dendrites of another neuron, but in some cases also with the cell body or even the axon of another neuron. And that is the basic overview of a neuron's structure and the essential role of each part. Now, neurons can be classified according to many of their intrinsic characteristics. One of the classifications that I want you to learn right now is the classification based on the number of neurites. Remember that neurites is the general term used to describe any process that emerges from the cell body of a neuron, whether it is a dendrite or an axon. With this in mind, humans have three main types of neurons. First, multipolar neurons. These are the most common type, and they have multiple processes coming out of the cell body, one axon and several dendrites. Multipolar neurons are typically motor neurons or interneurons within the central nervous system. Second, bipolar neurons. These neurons have two processes emerging from the soma, one dendrite and one axon. They are usually found in specialized sensory pathways, such as the retina, the olfactory epithelium, and the inner ear. And finally, pseudonipolar neurons, sometimes called pseudomonopolar neurons. These neurons have a single process that comes out of the cell body, but it quickly divides into two branches, one peripheral branch and one central branch. These are the primary sensory neurons found in dorsal root ganglia and cranial nerve ganglia. This classification is important because each neuron type is closely related to a specific functional role in the nervous system. Now, let's take a look at the glial cells of the nervous system, starting with oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. What we need to understand about these cells is that their main function is to provide a lipid-rich covering around neuronal axons. This covering is known as the myelin sheath, and its primary purpose is to greatly increase the speed at which nerve impulses are conducted. There is an important difference between oligodendrocytes and Schwann cells. In the central nervous system, a single oligodendrocyte can myelinate multiple axons at the same time. In contrast, in the peripheral nervous system, each Schwann cell can myelinate only one segment of a single axon. Next, let's talk about astrocytes in the central nervous system and satellite cells in the peripheral nervous system. Both of these glial cells act primarily as structural support cells. They help maintain the organization and stability of neuronal bodies within the neural tissue. However, astrocytes have an additional and extremely important role. They are key contributors to the blood-brain barrier. They do this through their end feet, which closely surround the blood vessels in the brain, acting as a selective filter that helps regulate what substances from the bloodstream can reach the neural parenchyma. Satellite cells, on the other hand, serve as supportive cells around neuronal bodies in peripheral ganglia, helping regulate the microenvironment of these neurons. Now let's look at microglia in the central nervous system and macrophages in the peripheral nervous system. These two cell types are functionally analogous. They serve as the immune surveillance and phagocytic cells of the nervous system. Their core role is to monitor the environment, remove debris, respond to injury, and help coordinate inflammatory processes when tissue is damaged, whether due to trauma, infection, or other insults. Microglia and macrophages also assist in remodeling and repairing tissue after damage, making them essential contributors to neural homeostasis. One important distinction is their developmental origin. Unlike other glial cells that arise from the neuroectoderm, microglia derive from mesodermal lineage during very early embryonic stages, specifically from progenitor cells that migrate from the yolk sac. Macrophages, meanwhile, share. Finally, let's take a look at the ependymal cells. These are the cells that line the internal cavities of the brain that make up the ventricular system. Along these ventricles flows the cerebrospinal fluid, or CSF, which plays several essential roles. It cushions the brain, helps maintain a stable chemical environment, and contributes to waste removal. Ependymal cells form a simple epithelial layer with cilia that help circulate this CSF throughout the ventricular system and into the subarachnoid space. Now, within this group there is a specialized subtype, the choroid plexus epithelial cells, which are responsible for producing the cerebrospinal fluid itself. We will explore these in more detail later on, when we cover the ventricular system as a whole. Now we're going to cover a few key concepts before moving on to the functional approach to neuroanatomy. The first thing I want you to understand is how we name groups of neuronal cell bodies and groups of axons in both the central and peripheral nervous systems. In the central nervous system, a collection of neuronal cell bodies is referred to as gray matter. This gray matter can be organized into different structures, as cortex in the cerebrum and cerebellum, as nuclei throughout the brainstem, cerebellum, or deep regions of the cerebrum, or as laminae, as you can see in the spinal cord cross-section shown in the image. In the peripheral nervous system, however, a cluster of neuronal cell bodies is called a ganglion. Now, regarding axons, in the central nervous system they are known collectively as white matter, and they are arranged into tracts. In contrast, in the peripheral nervous system, axons are organized into nerves. In the diagram, you can also see that peripheral nerves have several connective tissue coverings, and these are exactly what we will examine on the next slide. Here we have a cross-section showing the different components of a peripheral nerve. A nerve is made up of numerous nerve fibers, or axons. Each axon is wrapped by a Schwann cell, which provides the myelin sheath. Surrounding this myelinated axon, we find the first connective tissue layer called the endoneurium. Multiple axons, each with its own endoneurium, are grouped together to form a fascicle. Each fascicle is then enclosed by a second connective tissue layer known as the perineurium. Finally, several fascicles come together to form the nerve itself, and the entire structure is wrapped by the outermost connective tissue layer, the epineurium. These three connective tissue layers are essential because peripheral nerves lie outside the protection of the skull and vertebral column. Being more exposed, they are far more susceptible to trauma, compression, or stretching injuries. The endoneurium, perineurium, and epineurium together provide crucial mechanical support and protection, helping preserve nerve function. Now we finally arrive at the functional approach to neuroanatomy. To introduce it, we'll use this simplified diagram. The circle represents the central nervous system, any of its structures, and the rectangle represents the peripheral nervous system, such as a peripheral nerve. In general terms, this is how the nervous system works. First, we detect a stimulus through a sensory receptor. That receptor converts the stimulus into a nerve impulse, which then travels through a peripheral nerve as sensory information carried by an afferent, or sensory, neuron. It's important to note that the cell body of this sensory neuron is typically located outside the central nervous system. Once the signal reaches the CNS, the information is integrated and processed. The CNS then generates an appropriate response, which is transmitted through a motor neuron that originates within the CNS. This motor neuron carries motor information toward an effector organ so that it can perform an action. This is, in its simplest form, the basic functional flow of the nervous system. Of course, we will explore each component in much greater detail as the course progresses. Now we need to introduce another important concept. I mentioned earlier that everything begins with a stimulus, and stimuli can come in many forms: light, sound, taste, touch, smell, and so on. Because there are so many different types of stimuli, there must also be many different types of nerve fibers capable of carrying each specific kind of information to a specific region of the central nervous system. Likewise, particular regions of the CNS send motor commands back to the body through specialized motor fibers, each targeting a specific organ to produce a specific action. We'll cover all of this in much greater detail later. For now, what you need to understand is that nerve fibers can be classified based on the type of information they transmit. In general terms, there are afferent sensory fibers and afferent motor fibers. In the diagram, the fibers highlighted in yellow belong to the somatic nervous system, which we will discuss later. The fibers highlighted in green belong to the autonomic nervous system, which we will also examine in depth. Finally, the fibers without any highlight do not fall under either of these classifications, but don't worry, we will study them as well in their corresponding lessons. Now, to deepen our understanding of the somatic and autonomic nervous systems within the functional perspective, let's compare them side by side. The somatic nervous system provides both sensory and motor innervation to the skin and to skeletal muscle. Its sensory component allows us to perceive touch, pain, temperature, and proprioception. Its motor component enables voluntary movements as well as reflexive actions. In contrast, the autonomic nervous system contains only motor pathways, targeting visceral organs, cardiac muscle, smooth muscle, and glands. It is further divided into two major divisions. The sympathetic division, which promotes fight-or-flight activities and predominantly uses the neurotransmitters epinephrine and norepinephrine. The parasympathetic division, which supports rest and feed functions and mainly uses acetylcholine as its neurotransmitter. To give you some examples of how these two systems work, here are a few key comparisons. The sympathetic nervous system causes pupillary dilation mydriasis, while the parasympathetic system promotes pupillary constriction miosis. Regarding exocrine glands, the sympathetic system inhibits glandular secretion, except for sweat glands. In contrast, the parasympathetic system stimulates exocrine secretion. In the heart, the sympathetic system increases heart rate, enhances cardiac contractility, and accelerates conduction velocity, whereas the parasympathetic system produces the opposite effects. In the bronchi, the sympathetic system causes bronchodilation, while the parasympathetic system causes bronchoconstriction. In the gastrointestinal tract, the sympathetic system reduces peristalsis and contracts sphincters, while the parasympathetic system enhances peristalsis and relaxes sphincters. For the adrenal gland, the sympathetic system stimulates its secretion, while the parasympathetic system does not stimulate adrenal secretion. In the urinary bladder, the sympathetic system relaxes the bladder wall and contracts the sphincter, whereas the parasympathetic system contracts the bladder and relaxes the sphincter. Finally, regarding sexual function, the parasympathetic system promotes erection, while the sympathetic system is responsible for ejaculation. These are just a few examples of how both systems exert their effects throughout the body. If you found this helpful, please share your feedback. It helps me make future classes better. Follow my main Instagram for updates, neuroanatomy posts, and learning tips.